they specific to peroxides? And the reason that we asked this question was that it was well known in the field that mutants like mutants in the insulin receptor DAF2, in addition to living longer, they're also resistant to pretty much anything that you throw at them: arsenide, paraquat, producing agent, and DDT. So we asked, are DAF7 mutants resistant to all those other things too? And they're not. So you can see that when we expose them to a reducing agent, DDT, DAF7 mutants have identical survival as wild-type animals. When we expose them to a redox cycling herbicide, paraquat, also there's no statistical difference. And when we expose them to sodium arsenide causes metalloid, also can cause oxidative stress, also there's no difference. So DAF7 mutants are specifically resistant to peroxide. So we think that the reason that DAF7 regulates peroxide resistance specifically is that peroxides are really important to the worm. So the worms evolve as a dedicated communication channel from the neurons to the rest of the animal to regulate the animal's resistance to peroxide. And later on, I'll show you there's also other forms of peroxide like hydrogen peroxide, not just hydrogen. Okay, so how does this hormone DAF7 from this ASI sensory neurons regulate the worm's peroxide resistance? So there's a very well-studied TGF-beta pathway in C. elegans, works in many labs, including Don Riddle's lab, including Murphy's lab, the Rockman's lab, and Heidi Tissenbaum's lab. There's many others that I'm forgetting. They put together this TGF-beta pathway and this receptor called DAF1, and this cosmal called DAF3 that is repressed by the receptor. And sure enough, loss-of-function mutants in the DAF1 receptor also increase peroxide resistance in a manner that was mostly dependent on the DAF3's cosmal. So if we remove DAF3 and wild-type, and otherwise wild-type animals, there's no effect. But if you remove DAF3 on DAF1 TGF-beta receptor mutants, then most of the increased peroxide resistance. And importantly, this DAF3 dependence also occurred for this ASI ablation. So that told us that DAF7 was the major mechanism that mediates peroxide resistance from ASI. Okay, so how does this signal from ASI regulate peroxide resistance? So in the next couple of slides, I'm going to summarize a couple of years of work and hundreds of experiments by grad students and also by those 8 undergraduate students that know all these mutants, double mutants, triple mutants, quadruple mutants in the lab. They were amazing. And we were able to look at, do pathway analysis and also determine the site of action of 4 different genes along the way. And so what we found was that this ASI sensory neurons make this hormone DAF7, and that the reception of this hormone by the DAF1 receptor doesn't occur in target tissues to protect the animal. Instead, it occurs in multiple sets of interneurons, including the set 1. There's only a pair of interneurons called the ADK neurons, and this other set that includes the rim and brick interneurons, and this other set includes neuropharyngeal neurons. Now, these neurons function redundantly. DAF1 functions redundantly in these neurons. So if any of these neurons is expressing DAF1, they can respond to the DAF7 signal from ASI and tell the animal to have the normal peroxide resistance. But if DAF1 is missing from all these sets of neurons, then the animal induces its defenses. So we think that this system ensures that the animal only commits to inducing its peroxide defenses when all these interneurons are failing to respond to the DAF7 signal, and also doing tissue-specific rescue experiments of the DAF3 cosmal, we found that the opposite is true, that DAF3 functions in a non-redundant manner in these sets of neurons, and this is because there's an inversion in the signaling where the receptor is repressing. Okay, so how do these interneurons regulate peroxide resistance? They do it by regulating the expression of several insulin genes. And we found 6 insulins that were known from the work of Heidi Tissenbaum and Murphy's lab to be in the Patterson's lab to be regulated by DAF3. And these insulins, out of the 40 insulins in the worm, we found that 6 also regulate peroxide resistance. And now, So when DAF3 represses the expression of this insulin. So when there's DAF7 around, DAF3 is off and these insulins are re-expressed, and they tell target tissues through an insulin receptor DAF2 to repress two transcription factors, DAF16 and SKIN1. Now DAF16 is the one's ortholog of FOXO, and SKIN1 is the one's ortholog of NRF, the nuclear regulatory factor, and both of these transcription factors have been shown in many organisms to regulate many stress responses, including the oxidative stress response. We found that DAF16 and like with SKIN1, they function in the intestine to respond to these signals from the body. Right. So this signal from ASI hormone, and this hormone TGF-beta is relayed by interneurons to target tissues through the action of insulins, and these insulins repress transcription factors that coordinate transcriptional response that protects the animals from peroxide. And we actually found out that one of the genes that these transcription factors regulate is actually an enzyme catalase that degrades hydrogen peroxide and protects the animals from peroxide. Yeah, in fact, here's a slide showing the RNA-seq analysis comparing DAF7 mutants and wild-type animals. And the point here is that you remove 10 neurons, and there's about 600 genes that go up fourfold, and about 1,200 genes that go down fourfold. So it's a dramatic remodeling of the transcription in response to this signal from these two neurons. And this work is also consistent with previous microarray studies done by Colleen Murphy's lab, looking at the regulation of gene expression in response to TGFB signal in peroxide. And what you see here is like a kind of a clustering of gene ontology analysis in the semantic space. The point here is that we can look at these genes that are regulated, and they include genes, proteins involving in defense responses and responses, immune responses, and also in metabolism. And many of the genes that are downregulated in animals will reduce TGF-beta signaling and cause genes expressed in the germline involved in proliferation. So we're interested in how these genes work together to regulate the warm peroxide resistance, and we're also interested, and we're working heavily on this, on how other neurons regulate the transcriptome, where they converge with DAF7 and regulating the same genes, or they regulate different genes to protect the animal from this oxidative stress by peroxide. All right, so this is how this neuron regulates the warm peroxide resistance. These neurons and the signal essentially tells the target tissues to turn off the defenses. And so when is this important? Is this something that actually happens in real life? And now, work from many labs, including Corey Bachman's lab, including Cheng's lab, Don Riddle's lab, and Yang Yu's lab, together what they found with the DAF7, the expression of this gene in the ASI neurons is regulated by many signals, including food. And for worms, the food is bacteria. And so, in the expression of DAF7 goes up when the worms are well fed. And if you starve the animals, the expression of DAF7 goes down. If you refeed them, it comes back up. And also, the expression is mediated by sensory perception of food and these sensory neurons. And so you can see here GFP reporter for DAF7 from Andy Cheng's lab paper and Corey Bergman's lab, and you can see this green dot here is the ASI neurons, and this text for sensory transaction defective mutants that I'll tell you are more resistant to peroxide and also wrong. These mutants do not express this. All right, so sensory perception of E.coli induces DAF7 expression in ASI. So based on this line of evidence and other experiments that I haven't shown yet, we put up a model where E.coli perception induces DAF7 expression, and this leads to the animal to turn off its defenses. And so when there's no E.coli around, DAF7 expression goes down, and then the animals induce DAF16 dependent gene expression in the intestine, and that protects the worm from pests. So this makes sense when the animals can protect themselves by inducing their defenses when they have food. Why would they? And we actually found that food ingestion also was protected from peroxide. So it makes sense to us that when the animals consume food, then they induce their defenses. But why do the animals turn off their defenses when they sense bacteria? This was fascinating to us because it made the animals more vulnerable to peroxides, and also it's kind of reminiscent of this you know this thing that I told you at the beginning of the talk that worms have this latent capacity to live long, but they don't use it. That 16 is the major effectors of the insulin pathway, and it promotes increased lifespan. So, we really didn't have the answer for this question for a long time. It took us about 2 years to figure out the answer to this, and it really the answer came by really re-examining many experiments that hadn't, or we thought that hadn't worked. But when we look back and we talk to people, microbiologists that knew a lot about bacteria, and also people at the World Meeting, in particular Ilya Rubinsky, who really challenged us to really think, why would worms use their sensory neurons to regulate this phenotype? Now we now have a pretty good idea of what's happening. So it turns out that E. coli, so here you see like a plate with food where we put the worms and we drop in some hydrogen peroxide. You see all these bubbles. This is because E. coli is really good at degrading hydrogen peroxide. It's not so good at degrading other peroxides in the environment, like diluted peroxide. And that's why we've been using diluted peroxide because it's a stable peroxide. But in fact, so maybe E. coli was protecting the worms from hydrogen peroxide, and that's why the worms are turned off their defenses when they smell bacteria in the lab, E. coli. All right, and so in fact, we have been trying for a long time to kill worms with hydrogen peroxide, because I told you that hydrogen peroxide is the major chemical weapon of the microbial biosphere. But we couldn't kill the worms with hydrogen peroxide, even using these very high concentrations of peroxide that are clearly above what's physiologically or plausible. All right, so perhaps E. coli, by expressing enzymes that degrade hydrogen peroxide, like these catalases that make oxygen and cause bubbles that I just showed you about, by dismutating hydrogen peroxide without really needing any energy, which is really neat reaction. Perhaps this E. coli catalases is protecting the worm by degrading hydrogen peroxide in the worm's environment. So the worm, when it smells E. coli, turns off its defenses. This was rampant speculation, so we decided to do an experiment. And so we got this strain from Jim Inslee's lab. So we wild type E. coli and this E. coli triple mutant that is missing the two catalases, which are the two major enzymes degrading hydrogen peroxide in the environment, and also peroxide reduction. And so these bacteria are unable to degrade peroxide in the environment. And sure enough, when we do a survival assay in the presence of this triple mutant, then we were able to kill worms with hydrogen peroxide. Only 1 millimole hydrogen peroxide was enough to kill the worms in about half a day. And what we call we couldn't kill the worms, as we saw before, even when we added peroxide to the plate. And so these told us that these enzymes from E. coli were degrading the peroxide in the environment and protecting the worms from E. coli. So, in fact, these TAF7 mutants that were resistant to the diluted peroxide were also resistant to hydrogen peroxide when E. coli couldn't degrade. All right, so this altogether tells us that this signal from ASI, TAF7, enables worms to freeload of the hydrogen peroxide protection from E. coli. When the worms smell E. coli, they express TAF7, and the worms turn off their self-defenses. They turn them off. Why do they turn them off? Because they are expecting to be protected by E. coli, because E. coli expresses enzymes, catalases, reductions, degrade the peroxide from the environment and protect the worm from hydrogen peroxide. So worms don't need to induce their own defenses when they smell E. coli. Now, if the E. coli levels are low, then they turn off that same expression. This induces the hormone regulator I told you about, TAF16 localization in the intestine, and in turn increases the expression of defense genes, right? And that protects the animal from peroxide. So we want to know, okay, so if E. coli catalases are protecting the worm. The worms also use this. What are the mechanisms to protect the worms when the E. coli catalase are not protected? And so we speculate, well, perhaps it's the same gene. Perhaps the worm's endogenous catalase are protecting the worm from hydrogen peroxide when E. coli is not protecting. Yeah, so yes, so once I include 3 catalases in the genome, and we knew from our RNA-seq data that all 3 of them, or 2 of them, CTL1 and CTL2, which are major catalases present in the intestine, they're induced in response to lower TGF-beta signaling, and this is consistent with previous work from Colin Murphy's lab showing that the insulin pathway regulated the expression of all 3 catalases. And so what you see here is a reporter for this catalase 1, the major cytosolic catalase, and you can see that it's kind of dim in wild type animals, but it's much brighter in this type 1 TGF-beta receptor mutants, and this is the quantification of the level and consistency of expression. We did this also via qPCR in collaboration with Arjun Gassi's lab and also the RNA-seq. So we're pretty sure that these enzymes are induced in response to lower levels of TRAP7 expression. But this enzyme is important to the increased peroxide resistance of these mutants, and they are. So here is the DAF1 mutants, the TGF-beta receptor mutants, and the represses one as wild type animals in the presence of hydrogen peroxide when there is this E. coli strain that can't degrade peroxide, right? And these catalase 1 mutants live a little bit shorter than wild type animals, but they live a lot shorter when in DAF1 mutants. So these double mutants with about 50% shorter, or 50% of the effect of DAF1 is mediated by the CTL. Right. So this tells us that the increased expression of catalases in response to a downregulation of TGF-beta signaling from this ASI sensory neuron is protecting the animals from hydrogen peroxide. In fact, if we overexpress catalases in the worm, we can increase peroxide resistance further. Right. So altogether, we come up with this model. It's very simple. That C. elegans is really always ready for the threat of hydrogen peroxide. When there is no food around, then the 7 gene expression slows down, and the worm induces its own defenses, including this cytosolic catalase. But when the worm senses bacteria, then the worm turns off its defenses. But this doesn't mean that the worm is not prepared. The worm is prepared because the worm, we think, is expecting the E. coli to protect the worm from hydrogen peroxide, because E. coli expresses these enzymes that degrade peroxide. In fact, it's the same gene with catalase. So the worms are using really the brain to decide whether to express their own catalases or to rely effectively on the catalases from. Okay, so is this something that happens only in worms, or does this happen in other organisms? So it turns out that this cross-kingdom hydrogen peroxide protection really is everywhere. So in fact, so if you look in the ocean, the major photosynthetic organism is cyanobacteria, Prochlorococcus, and they don't really have their own catalase. In fact, you cannot grow cyanobacteria in the lab unless you really get rid of every little last trace of hydrogen peroxide on the plate. If you could culture them with bacteria that degrade peroxide, most bacteria, then they will fine. So actually in the open ocean, bacteria that express catalases, they degrade peroxide and protect cyanobacteria, and this is why these organisms were able to remove the atmosphere of producing so much oxygen. But also diatoms are closely associated with bacteria. So we think that this is going to be an important principle, that this cross-kingdom peroxide protection. Right. So why don't worms just turn on this system all the time? Why regulate catalase gene expression? Why not just turn it on all the time and always be ready? Why have a brain regulator? We think that expression of catalase is very costly. And so, in fact, this beautiful work from David Jen's lab showing that if you overexpress the catalases, the worms are kind of sick. They develop more slowly. And here's a drawing by a student from David Emily Underwood. And this is what happens to a worm that expresses catalases. They form these bag of worms. They are so sick, they don't lay eggs, and the eggs hatch inside the worms, and they kind of eat their way out. So it's clearly costly for the worm to express this gene when it's not needed. All right, so now this DAF7 gene that regulates, just told you the reverse peroxide resistance, has many other functions in the worm. It promotes the formation of these stressors of tower larvae, it controls fat storage, germ growth, laying, feeding. So we actually think that we can reinterpret the functions of DAF7 in light of what we found out on its effect on peroxide resistance. So all the phenotypes that DAF7 is regulating are actually consistent with a coordinated broad defensive response to the threat of hydrogen peroxide. So in the presence of hydrogen peroxide, or when if you're worried, you're not expecting to be protected from peroxide, it makes sense to inhibit feeding. Peroxide is toxic. It makes sense to inhibit egg laying, because why lay an egg if your egg is going to die in the presence of peroxide? Inhibit germ growth and so on. Right. So why do sensory neurons make the worms age faster? And so we think we have a good answer for this. And our answer is that sensory neurons make worms age faster because they turn off the body's defenses when the neurons in the worm are expecting the worms to be safe from hydrogen peroxide attack. So if the animal doesn't have DAF7 or is missing the ASI sensory neurons, they induce a protective response that increases lifespan. And this is beautiful work from Joy Alcero's lab and also from Colleen Murphy's lab, showing that these sensory neurons specifically regulate longevity. But this happens in the absence of stress, right? So what we think is happening is that the animals normally, when they sense food in the normal conditions that they run in the lab, they shut down these defenses via this TGFB-insulin hormone relay because they're expecting to be safe. But if we can trick them into thinking that they're not safe by impairing sensory perception within ASI or other sensory neurons, then the animal can induce protective defenses, including these catalases that protect the worm from hydrogen peroxide, and but also the STAF16 dependent gene expression, also dependent gene expression that can induce defenses that can increase lifespan when the worms are not in the presence of stress. All right, so perhaps sensory neurons in our body are regulating our own lifespan. Perhaps we can trick our own bodies into thinking that we need to induce these defenses and they may regulate our own lifespan. In fact, sensory neurons, pain sensory neurons in mice regulate immune responses, including whether immune cells produce hydrogen peroxide and also whether somatic cells induce defenses. So we think this is going to be possible. All right, so now I'm going to switch gears in the last 15 minutes and tell you about how sensory neurons in the brain regulate avoidance of bacteria that do not provide hydrogen peroxide protection. And this was work led also by Jody and an amazing undergrad, Stephanie Steiner. So they're going to be, actually it's in press now. It's in bioRxiv if you want to read it, but it's in press and it's going to be published too. All right, so the question that we got started was, can worms tell the difference between bacteria that provide hydrogen peroxide and bacteria that do not, or are they just guessing, right? And so to answer that, what we did was to do a choice assay. We put worms on a Petri plate, and we put two bacterial lungs. One, the E. coli bacterial lung, the one type, and then here in red, this E. coli, the triple mutant lacking the catalases and the peroxide redoxin, so bacteria that can degrade peroxide. Then we put the worms in the middle and we say to the worm, okay, go. And we waited some time and we watched where they went to the one type E. coli or to this catalase effective E. coli here. And we've done this many, many times. And here are some representative pictures of what you see. And you can see that the worms, as a function of time, they find these lungs, they find both of them, and in about equal proportion. In fact, they have a slight preference for the wild type E.coli. So they go about 60% to this one and 40% to this one. And we've done lots of quantifications. I'm just showing you the picture. And so in the absence of peroxide in the environment, the worms have a slight preference for the wild type E.coli. Now, if you put them in the presence of peroxide, something very dramatic happens. So early on, the worms go towards both the wild type and the mutant E.coli. But then you can see that these worms here are kind of they didn't stop at the edge. They kept moving around. After 30 minutes, they're starting to leave. And after an hour, you see far fewer worms at the log here. So they left. And then after 2 hours, very, very few worms stay here. Most of them made it to the end. So initially, they go to the fourth bacteria. Then, if the bacteria don't degrade the peroxide in the environment, the worms turn around and leave. So this is really amazing because, worms are really good at finding bacteria using a sensory perception because they need to eat, right? They need bacteria for growth and reproduction. So how are the worms overriding their strong attraction towards bacteria so that to leave this one? So to answer that question, we collaborated with Dr. Catalases lab and Northeastern, and what we did in collaboration was to watch worms using a microfluidic device and in worms expressing GCAM in 28 sensory neurons, 14 pairs of sensory neurons, including the major sensory neurons in the amphitheater, the main sensory organ in the brain. And what we did was to expose the worms to hydrogen peroxide or to bacteria and hydrogen peroxide, to bacteria that don't degrade hydrogen peroxide, to bacteria that don't degrade hydrogen peroxide and hydrogen peroxide. Altogether, 6 combinations. And watch how the dynamics of this calcium sensor in response to it. And what we see here, what we saw was really beautiful and summarized here in this heat map. And so the point here is that we saw 2 classes of responses. Some neurons responding to hydrogen peroxide, and they also respond to hydrogen peroxide when bacteria couldn't degrade the peroxide, but these neurons didn't respond to peroxide. Other neurons responded to food, but not to hydrogen peroxide, and also they didn't respond to food when there was peroxide around. And other neurons had they were inhibited by food, but not when with peroxide. Right, so let me show you some representative of what it looks like when you look at these traces. So here you're plotting the change in calcium or in the signal of the GCAM sensor when the worms encounter the stimulus, in this case 1 millimolar hydrogen peroxide. You see this huge response, and either just the peroxide alone or the peroxide and the bacteria that don't degrade peroxide. But if you just expose them to bacteria alone or bacteria that degrade peroxide, then you don't see anything. And these other neurons, these ADF neurons, and many other neurons that respond to food, that respond beautifully to food, but not when this hydrogen peroxide and the bacteria can degrade it. So these ADF sensory neurons work from Aaron and Levin's lab. These neurons are neurons that respond to bacteria, and they help worms slow down as they encounter bacteria. So there are 2 classes of neurons that respond to peroxide and neurons that respond to food, but the neurons that respond to peroxide don't respond to peroxide if the bacteria can degrade the peroxide, and the neurons that respond to food don't respond to food if the peroxide is there. Right. So now these SJ sensing neurons work in many other labs, have shown that they mediate many aversive responses. They respond to nitrous oxide from work on Kaplan lab, and then Skim's lab showed that they're involved in the avoidance of thiocyanins from Pseudomonas aeruginosa, and these other labs have shown that Chasani lab and I show that it's involved in avoidance of pathogen predator-derived cephalitis. So these neurons are excited in response to harmful things to the worm. So we wanted to know, and they are also excited in response to hydrogen peroxide. So we wanted to know if they are important for avoidance to peroxide. So what we did was to do this drop avoidance assay. So essentially, we put a drop of peroxide and we let the worms crawl into it. And if the worms don't like it, then they kind of turn around and make this what's called a megaturn and avoid it. So we count how often they make an avoidance response. And worms sometimes they avoid water, but they avoid the peroxide much more frequently. And now animals without these ASJ sensory neurons, because of the genetically ablated, they don't see this increase here. So in fact, there's no statistical difference. So ASJs require to avoid hydrogen peroxide, and it's also hydrogen peroxide sensitive. So together we can put up a model for how the worms decide when to stay and when to go. And so there are two classes of sensory neurons that respond to hydrogen peroxide, including the ASJ sensory neurons, but also the URX neurons and the ASK neurons. These neurons mediate avoidant responses. So far, we only know that ASJ is required for avoidance to hydrogen peroxide. Now, bacteria induce modulate calcium levels in many sensory neurons, and these are required for attractive behavior. But hydrogen peroxide can block bacteria from exciting those neurons or modulating those neurons. And we're still trying to figure out what is the mechanism. Where does hydrogen peroxide act to block the effect of bacteria on sensory perception? That's going to be a really exciting new direction for that. And the bacteria can also degrade hydrogen peroxide, because bacteria can have enzymes that degrade peroxide in the environment. And so you see bacteria inhibit peroxide inhibit bacteria. This is a cross attenuation. And this type of cross attenuation circuit essentially causes like a switch-like response. And you think that this switch-like cross attenuation circuit is helping the worms switch between avoidance and attractive behavior. And this is how the worms override its innate attraction towards bacteria in the presence of hydrogen peroxide when the bacteria cannot degrade the peroxide. Right, so the emerging theme is that C. elegans process sensory information to choose when to induce its defenses. And I told you today that the signal from ASI controls when to protect yourself if you're alarmed, when to induce your defenses. And these other signals from other sensory neurons control when the worms escape. So this escape behavior response occurs in a very short time scale, right? Time scale of seconds. This response when to induce includes a hormone relay and many transcriptional steps. So we think it's slower. And actually, part of what we're trying to grow and questions that we're trying to study next is to understand how this behavior and physiology, how they coordinate with one another. Because you can imagine there are many strategies that the worms could use to deal with the threat of peroxide. The worms, if they sense peroxide in the environment, could just avoid it and leave, or they could induce their defenses as they leave, so they could be prepared for the next time around. Or if they induce their defenses, they could decide to stay in the presence of this dangerous environment, because if there's food around, that might be better than leaving and never finding food. And so we want to study how these mechanisms that control when to protect yourself and when to escape from the threat, how they're coordinated relative to each other, and maybe they're polymorphic in the wild and different isolates of cells. And that's kind of what we want to ask next. So these are our big questions. How does the information from many sensory neurons is integrated and uses specific defense towards peroxide? How are the choices to escape or resist or repair the damage from peroxide? How are they coordinated? And how does aging and reproduction moderate those choices? So if you're interested in these questions, send me a message or an email, or reach out to me. We're looking for new grad students to work on these projects. Right, and last but not least, most important slide of all, to thank you to my lab, because nothing in this talk would have been possible. We have a lab, which are amazing. I'm so proud of them. And so here's a picture of the lab trying to troubleshoot the light machine. And so this work was done by Jody, Frank, Yuyan, and a new grad student, Maria, in the lab. And we have many undergrads that have worked on this project, many REUs, and we have many collaborators. I want to particularly thank Nick Straubstrup at the CRG and Vivek Venkachetalam. And I didn't really get a chance to talk about our collaboration with Buck Samuel and Michael Donald, looking at the role of microbiome in controlling whether one stay or leave in the presence of peroxide. We have other collaborators in other projects, including Felix and Gupta, looking at the role of the AFB, temperature sensing neuron. And I'll stop and take questions. And if you have some time, take a picture of the QR code. It will take you to forums to give us feedback. Okay, thank you.
Great, thank you very much, Dr. Apfeld. That was a fantastic talk. It's really comprehensive, and every time I kind of had a question come up in my head, the next slide would be the answer to the question. It's really impressive. So we got lots of questions, so we'll try and get through as many as we can. But as I put in the chat, if anyone wants to chat more with Dr. Apfeld and with a small group of us afterwards, there's a Zoom link there where you can join us for a more informal meeting. But yeah, so the first question was on the very beginning part of your talk, where you were looking at different mutants and affecting different sensory neurons and their cost in terms of their cost or benefit in terms of resistance to peroxide stress. So the question is which of those, or whether any of those mutants also have a cost in the absence of oxidative stress.
So,
and you kind of touched on that at the end of your talk. But,
yeah. So we look at different types of mutants. We look at some mutants that have broad effects that affect many sensory neurons, and those mutants are very long-lived. And we also look at these transgenic animals that genetically ablate specific neurons. We didn't really look systematically at the effect on growth and reproduction. That's a really interesting question. But we know from the work from David James' lab and also our own experience working with transgenic animals expressing catalases, that expression of catalases is sufficient to make the animals sick. And so, when you turn off TGA-beta signaling, you induce the expression of catalases and thousands of other genes. Now, some of those genes may offset the toxicity of the catalases. So by themselves, the catalases, when expressed, can be perhaps more toxic than when you have a coordinated response. I'm not sure if I answered the question.
Yeah, that's great. Thank you. Then another question here is whether there's a threshold for each kind of stressor, and what is the biological relevance in terms of what kinds of stressors C. elegans would be likely to encounter in nature.
So that's a very interesting one. So okay, so we looked when I show you, the slide that the DAF7 mutants are more resistant to peroxides, but not to other stressors. We tried to use concentrations of those stressors or those chemicals that would kill the worms in roughly the same time scale as the peroxides were killing them, around a day. These things are, sometimes, not exactly the same, but within 50%, they're in the ballpark. And so we think that we're using comparable concentrations. We haven't looked systematically to see whether higher or lower concentrations of those chemicals, there might be some effect by mutants in the TGA-beta pathways, or maybe there's different price points for different stressors. That would be really interesting. I think one important question for the field is to try to understand how these neurons regulate a specific stress response, in this case to peroxides, while the downstream effect of the insulin pathway regulates resistance to a much broader response. And we don't have the answer for that, although we have some ideas.
Mm-hmm, mm-hmm. Then an interesting question here about whether the downregulation of defense, peroxide defense, requires actual or live bacteria, or just the ability of C. elegans to sense the presence of bacteria. Like, what are they detecting about the bacteria to regulate defense?
Yeah, so we can look at live versus bacteria. But one thing we did was to expose the worms to different concentrations of bacteria before the assay. And what we found was that animals that were exposed to lower concentrations of bacteria or lower levels of bacteria, they live shorter. And if we impair the animal's ability to feed, these feeding defective mutants, these T2 mutants, actually live long under normal conditions. These more dietary restriction. These mutants live also short in the presence of beta peroxide. So ingestion of food increases peroxide resistance, but these DAF3 mutants that don't have this COSMAB downstream of the 7 receptor, the 7 hormone, when you remove DAF3 in the E2 mutants, they live much shorter. So this told us that this TGA beta pathway was induced in response to reduced feeding. So at least we know that the level of food ingestion is important, but we didn't look at, it's also possible that the DAF7 signaling may be regulated not necessarily by only the perception of food, but also by the level of food ingestion or by the quality of the food. Like, is the food alive or dead? Is it better quality in a different way? And we don't know that. That would be a really interesting direction to take this.
Mm-hmm. Yeah, and then two last questions. Firstly, does on the last part of the talk now, so does the preference for peroxide, for bacteria that break down peroxide, change with age?
We don't know. Great question. Yeah, it'd be great to look at other animals and also different concentrations to see if the pattern is different.
And then lastly, do you know whether ASJ neurons, which regulate that preference, also affect lifespan?
Well, that's already known. There's so the answer is yes. In our hands, those neurons not only promote avoidance to peroxide, but they also promote survival to beta peroxide. And this absolutely beautiful work from Joya Alcedo back when she was a postdoc in Cynthia Kenyon's lab. She did ablations of different neurons, sensory neurons, and different combinations of sensory neurons. And she found that the ASJ sensory neurons were required for the increased lifespan of animals where other sensory neurons, like the ASI sensory neuron, were ablated. And so it's like a neural code, like combinations of neurons work together to regulate lifespan, including ASJ.
Yeah, interesting. Great. Well, thank you. If you have more questions, you can always email Dr. Apfeld, or please do join the Zoom session, which will start in just 2 minutes or so, right after we finish here. And other than that, I'd like to thank again Dr. Apfeld for joining us this week. It was a really fantastic talk. And I hope, as I said, this is the last seminar of the year, so I hope many of you will be able to join us next year. And you'll, if you're not signed up to our email list, you can sign up to that on our website, and you'll hear about the next season of talks there or on Twitter, etc. So have a great end to the year, and we'll see you all next year. Thank you again, Dr. Apfeld.
Thank you.